Well, good morning now. We're going to start. Thank you very much for joining us. And we're going to be videotaping this panel. So for your questions at the end of the panel, please use the microphone. Welcome to the One Health Fall series seminars. And since I have a very heavy accent, I will try to speak very little. And I will ask our panelists to please introduce themselves. I think you have, all of you have a bio and an agenda and also a survey that if you could kindly complete at the end of the panel, that would be very helpful for us. So I will start with Dr. Scorza. If you could please briefly introduce yourself and your name, institutional affiliation, and why did you choose to be a veterinarian?
Well, my name is Valeria Scorza. I am a veterinarian. I work at CSU in the Clinical Science Department at the Center for Companion Animal Studies. And I wanted to be a veterinarian like pretty much every other veterinarian because we care so much about animals and their health. And that's why then I end up working with in the small animal lab.
Thank you. Dr. Abel, please, same introduction.
Sure. I'm Greg Ebel, and I'm a professor in the Department of Microbiology, Immunology, and Pathology in the College of Veterinary Medicine and Biomedical Sciences, and I'm also the director of the Arthropod-Borne and Infectious Diseases Lab, and I'm an arbovirologist, and that means that I work on viruses that are transmitted by mosquitoes and ticks.
Thank you.
And
Dr.
I'm
Olson.
Dan Olson. I'm from the University of Colorado, and I do pediatric infectious diseases and epidemiology. And so I've been kind of doing global health work for a number of years, but you'll hear a lot of the work we've been doing at our site, which goes back about 7 years. And I was thinking the other day, if I had to pick a different career from what I did, I would either be a veterinarian or a virologist. So we're a good team here.
Wonderful. Well, welcome. And Dr. Scorza, I think you're starting?
No.
No, Dr. Olson.
Yeah. I'll just stand over you. Okay, so I'll just talk really loud. So what I want to do is kind of summarize 7 years of human research that we've been doing at our site in 15 minutes. So this is going to be like a rapid fire bird's eye view of some of the projects, and I'm happy to kind of go into more details as we kind of have questions and things like that, okay? So first I'm just going to briefly mention our site. This is called the Centro de Desarrollo Humano. It's in the Center for Human Development in this area of Guatemala.
Dan, I'm so sorry to interrupt you, but
You want me to use the microphone?
Could I have you hold the microphone?
I'm
I'm just checking with our video, and I want to make sure the audio gets picked up.
Okay,
I
yes.
apologize for the
No,
interruption.
no problem.
Thank you.
So our site is located in the southwest Trifinio region, it's called, because it's where 3 different health districts come together. It's right along the border of Mexico, and this is actually where the migrant caravans come through, so it's kind of like a strategic area from population migration standpoint. Here's some pictures of our clinic. The site was originally founded as a foundation, NGO locally, and it had financial support from the University of Colorado, but also from a different foundation locally, which was run by a large banana plantation. And the original focus was on healthcare to the communities, focusing particularly on the workers for these big banana plantations, but also their families and the community at large. And so the first step was this clinic that's on the bottom left here. There we go. Oh, it doesn't show up on the screen though. So providing clinical care, but we've also managed to develop the site significantly, focusing more on research, and now we have a little entomology lab and other areas. The first thing we did was conduct a community needs assessment focused on what are the needs of this large community, about 200 square kilometers. And a couple of interesting statistics that we found from this survey was, one, only 52% of the population completed 2nd grade. 47% reported food insecurity, and more than 40% of kids were stunted. The under 5 mortality rate was pretty significant. And as you can see, a lot of the infrastructure that we take for granted in the US is pretty minimal there. So there's an example of how people get their water in the area, and only 60% of the population use a shallow well to collect water, which we could talk a little bit more as a risk factor for having diarrheal disease because the water gets contaminated. Our first big program in the area was really focusing on maternal and child health, and it's called Creciendo Sanos. Founded in 2013, we've now enrolled over 3,000 mothers and children. And the focus is not on research, it's on the health of these mothers and children when they get through this very risky period of pregnancy and early childhood. And includes multiple visits at our clinic and at the home, doing groups in the community. And we offer things like ultrasounds, but really what we focus on is kind of identifying red flags that are warning signs that these people are not doing well from a health standpoint, but also focusing on neurodevelopment and stimulation for the kids. And so we've had a couple of papers here that I'm happy to kind of tell you about. Our first big research study was in 2015 and 16, and the goal of this study was to identify cost-effective ways to estimate the burden of disease in very resource-limited settings. As you can imagine, knowing how much dengue or norovirus there is in a community is really important if you want to decide about introducing a vaccine or doing some kind of public health intervention. But traditional surveillance systems are actually pretty expensive and not always very accurate. And so we studied two different approaches. One was we enrolled 210 households. We gave each household a really cheap Android phone that had a smartphone app on it. And they would be responsible for reporting each week if anybody had fever or diarrhea. And if they did, we'd send somebody out to the home and collect a sample and ask some more questions. The second study was just doing rapid cross-sectional surveys where we'd go out into the community, do testing and interview questions on a lot of 210 households, about 400 kids, and find out what pathogens they had and then what are the risk factors they had for having these pathogens. And we did 2 of these cross-sectional surveys. And then, of course, we banked all the remaining samples, and we've tested for dengue and norovirus originally, but then Zika also came to the site during this time period. So we also looked at Zika. So some of the more interesting results from that. First off, we found, not surprisingly, a very high burden of diarrheal illness and norovirus, which is one of the common causes of diarrhea. We found a high burden of dengue, which I'll kind of go over here in a second, and Zika. And we also identified lots of risk factors for these diseases. Some of these are modifiable things that the public health system could do to kind of reduce the burden of disease. This shows you, this is a slide that I'll be presenting in a couple of weeks at a tropical medicine conference. I think the main take-home points here is this top part is what we found for our dengue testing, and the bottom part is Zika. Kind of the main take-home points is that the dengue prevalence, so this is how many people had dengue at some point in their life, was anywhere from 50 to 90% of the kids, depending on what test you use to determine that. So a very high burden of dengue in this pretty young population. These are all kids, so we've had lots of dengue in the community. And then the interesting thing here is for Zika, the prevalence, which is in the parentheses here, what proportion of kids had Zika antibodies, anywhere from 1 to 13%. And that shot up to almost 40% just during a 3-month period when Zika was first arriving. And so that's kind of an impressive change in just a couple months. And I think it speaks to how kind of fast Zika spread through Latin America. And so now we're going to be looking, we also have some stuff on risk factors and things for acquiring Zika. And this is an example of kind of putting yourself in a position and then taking advantage of, so this was a dengue study. Then all of a sudden Zika came through, and we were able to learn a lot about Zika just from the samples that we had already collected. And those biobank samples are really helpful for that. The last thing here I just wanted to show, so this smartphone study, we basically enrolled this group of kids. Every week they'd tell us if they had a fever using the smartphone app, and then we would go and test them for dengue or norovirus. This shows how many people sent us reported in a given week. And so it was around 80% overall, which is pretty good for anything where someone has to self-report every single week. And of course we reminded them. But I love this part. This is what happened when we had a big drop-off in reporting. And the first thing was we had a big staff turnover. At one point, a cell phone tower collapsed in the community, and so the reporting dropped because people weren't getting signal. And then Guatemala always has very contentious elections, as you may have heard in the news. And so during each election cycle, we had a big drop in self-reporting because there were protests and blockades on the roads and things like that. And of course, we also do a lot of GIS mapping in the area, so we can tell where are the areas that seem to have more dengue or Zika or norovirus based on these GPS coordinates that we've collected over time. And here we have a couple of papers. And by the way, I'm happy to send you guys papers on any of this stuff if you have more interest. This one I just wanted to mention briefly. This is a study where we looked at kids with moderate diarrhea. They would come into our clinic, and we would do what's called multiplex PCR, where you can look for 26 different pathogens at once using just a PCR test, looking for little bits of DNA. And the average child with diarrhea had almost 5 different pathogens in their stool. And we're talking about things that really cause diarrhea, like Shigella, E. coli, Giardia, Campylobacter. The average kid without diarrhea, the control group, had between 2 and 3 pathogens in their stool. And so that a normal US kid would not have any pathogen in their stool, and if they had diarrhea, they just have 1 usually. So this kind of speaks to the burden of enteric disease in this community. They're constantly exposed to stuff they shouldn't be exposed to. And it gets back to these wells here. Whenever it floods, the open pit latrines kind of flood into these wells and contaminate them, and that's why everybody has diarrhea all the time. And then our most recent study, other than the xenosurveillance study, which Greg and Valerio mentioned, is this pediatric Zika study where we looked at, the background is we know fetuses have a hard time when they get infected, exposed to Zika, but what happens when young children get exposed? Their brains are still developing and they may have problems as well. So in this study, we enrolled 500 infants and their mothers, as well as a bunch of older kids. Some of them were in this prior dengue study. And we did active surveillance for Zika to see if anybody had a Zika infection. We also looked at dengue and chikungunya. And then we did very intensive neurodevelopmental testing on these kids, which I'll tell you a little bit about. The neurodevelopmental testing was interesting. We had to adapt some of these tests to this very local, resource-limited setting. Some of these tests ask about, ask the kids about sledding. Like, do you know when's the last time you went sledding? Which obviously wouldn't be something that most kids in rural Guatemala would be exposed to. And so we had to adapt this Mullins test to the local context and kind of do a site validation to see how well it worked. And this was supervised by PhD neuropsychologists at University of Colorado, but was run primarily by local psychologists who really understood the local setting. And so this really this was the first huge study we did at our site. We had a really big research team and really kind of upped our capacity to kind of do bigger studies. And we have collected thousands of samples from this. This is just kind of goes through some of the tests we did, but suffice to say, it was a lot of work. The psychologists did over 2,000 Mullins tests. Each one is 30 to 45 minutes. So you can imagine the amount of work that went into doing this. For surveillance, we did what's called, we wanted to follow kids and identify kids that have what we call a flavivirus-like illness. And so that was comprised of the symptoms shown here below. And this shows what percentage of people that had a fly had each of these symptoms. So fever was really common, rash was common, and then some of these other symptoms were a little bit less common, but more so in the mothers. That's cohort M. And then again, we were able to satellite, track the cases of Zika as well as poor neurodevelopment using the satellite mapping here. The red is either Zika positivity or poor neurodevelopment. And those really tended to cluster in the more rural areas, which isn't a surprise. This is kind of the most important result from this study. And here, this is the results for age on the bottom and the developmental testing. And so this blue line here is the composite score. So this is like the overall score from this Mullins test. The normal score is 100 in the US for a US-based child. And each one of these is a subdomain score. And these ones are all normalized. So a normal score should be around 50. As you can see, these youngest 2 age groups, their overall score was pretty close to 100. So not a lot of delay. They're getting breastfed. They're not being exposed to some of these pathogens as high as some of these older kids. But look what happens when they get to around a year, between 1 year and 2 years of age. Their overall score plummets, and so do the subdomain scores. And the average kid in this study, their developmental score was 2 standard deviations below what a US score is. So we're talking 2.5th percentile was the average score for a child enrolled in this study. And so that really is impressive. And it's not because these kids were exposed to Zika, it's because they've been exposed to malnutrition and anemia and lack of stimulation, and all these other problems that we don't really get into too much depth in a lot of these settings, although when you talk about the overall impact on these kids and their development, their life, these are the things that really have a huge impact and even an economic impact. And then finally, I just wanted to mention, in January, we're going to be starting up a study on these agricultural workers. This is going to involve around 3,000 banana workers, and we're going to look at the impact of influenza. And so, right now, we don't really know how much flu there is in a lot of these populations in Latin America, especially in low resource settings. And one of the reasons why flu has become important and vaccine has become important in the US is not just clinical, although that's important too, but also economic. Things like work absenteeism, lost productivity, healthcare expenditures, those are all reasons why we vaccinate on a public health level in the US. And so for this study, we want to follow these workers and find out when they have flu. But these banana plantations, they actually pay their workers based on their productivity. So how many bananas did they pick in a given day? And so when we identify a case of flu, we'll be able to show how much absenteeism the workers had and also how much their productivity dropped. Then in the second part of the study, we're going to do a vaccine intervention where we give vaccine to the workers. And in the end, I guess my own personal goal is we'll be able to demonstrate that the burden of flu is high enough, the economic burden is high enough, that these workers, that the company actually would benefit economically by providing vaccine to the workers. Right now, flu vaccine is pretty much unavailable in these settings. No public health system is going to pay for annual flu vaccine when they have bigger problems. But if you could convince a company to provide flu vaccine to their workers, that would be completely outside of the normal Ministry of Health vaccination program. It would save lives for sure. And that could be done for lots of these diseases we talk about. Chikungunya would be another example where they develop this kind of chronic arthritis that really impacts their ability to work. So when a chikungunya vaccine comes out, that might be another example of an approach you could take. These figures just show how they track productivity, and these are individual workers and how much they get paid each day. And so you can see that changes quite a bit, especially when they're absent. And then one other point was that these workers have really harsh working conditions, and there is this thing called Mesoamerican nephropathy. Have any of you guys heard of that? So that's a disease that's seen commonly in agricultural workers in Latin America, especially sugarcane workers, where they spend so much time out in the field that they get dehydrated and then they take injectable ibuprofen. Those things combined really kind of hit your kidneys hard. And so a lot of these people actually have pretty significant kidney disease, which is a risk factor for severe influenza. And there's some researchers at the Anschutz campus that really are focusing on this whole Mesoamerican nephropathy. The average worker needs to drink 20 liters of water in a given day in order to stay hydrated. So you can imagine how hard that is to maintain hydration. And this last slide just shows a couple of other projects that I wasn't going to go into. The one I wanted to mention was this one that we started last year, this design hack course through the Department of Engineering. And this course, we bring 8 to 12 students down in the engineering school, although we're going to eventually open it up to public health students too. They come down to our site. These are undergrads or also first-year graduate students, and they partner up with these local agricultural students in Guatemala, and we do what's called a design hack competition, where each team has to identify a public health problem, like diarrheal illness, for example, and hack a low-cost solution to that problem. And then we have a competition, and the winning team gets a couple $100s to try to implement their idea. So last year was the first year we did it. Now we're doing it every single year in January. And we have a couple other projects we're working on, which I'm happy to talk about also. So that's it for me. Here's some more shots of the site. It's really awesome. We want people to come down there and do research, and happy to talk about more, I guess.
We're working on putting together a memorandum of understanding One
Yeah.
Health
Right.
for you, so
Yeah.
there's opportunities in the future.
Yes. And I hope this has been a focus on human surveillance, but I hope it kind of gives you an understanding of why One Health is so important in this area. I mean, these guys will talk a little bit more about that, but animals and mosquitoes are everywhere, and you can't just study these people in isolation. So we're really excited about partnering with the One Health Institute and kind of doing more collaborative work. So,
Thank you.
All right, well, that's it for me. Next.
Okay, so got to figure out which hand goes with which thing. All right, so I'm going to start today by, so Dan told you about kind of the human-focused studies there. I'm going to tell you about what we're doing with mosquitoes and introduce you to this concept of xenosurveillance. I'm going to tell you kind of what the premise is, kind of why we got interested in it, and then this is going to also be a little bit more lab-focused, and that I kind of want to tell you about some of the molecular biology that we've done to try to make it work. So it's like a cool idea, but then you got to figure out how to actually do it. And so I'm going to tell you a little bit about that and how we're implementing it, and then hopefully provide an introduction to the current One Health project that Dan and Valeria and I are working on. Okay, so the idea behind xenosurveillance is that you go to a place like El Trifinio in Guatemala or Africa and throughout much of the world's tropics, and mosquitoes are biting people all the time, every night. And so the idea is that those mosquitoes, when they sample human blood, are basically acting like little autonomous blood collecting devices. And there's blood in the mosquitoes, and there's information in that blood. And so our idea is that if you use the mosquitoes as blood sampling devices for you, when you go and try to do surveillance in somebody's house, instead of asking them to stick out their arm or prick their children's finger and making everybody sad and unhappy, instead of doing that, you go in and you're removing mosquitoes from their house. And so that sort of changes the whole ballgame. Okay, so when we first started talking about this, we were talking about it as a method for doing surveillance for new emerging infections. We were pitching it to the military. And the Department of Defense understands things like explosions and epidemics. And so we were talking about this as a concept of left of boom. And so the idea is that if this here is like your explosion or your outbreak, what you want to do is know about this, not up here or over here. You want to find out about it somewhere back in here. And so if you think about this in the context of an outbreak, or the emergence of a new disease, say something like Ebola. So the virus is out here all the time. It's just because there's no outbreak doesn't mean it's not there. But then as time goes forward, and this is time going on this axis, and cases are over here, there are a few cases that occur kind of, there's this little sort of chatter back and forth between nature and people. And so people are getting infected, and then finally you sort of make this jump from being a strictly zoonotic infection to one where there's a few people that are infected, and then you get this human chain of transmission, and then suddenly there's this epidemic. And so the idea is that here's the zoonosis phase, there are these initial spillovers, you have this emergence, and then here's the epidemic. Okay, so if we wanted to do like the virus hunter approach, which maybe you've heard about, we go out to nature and we sample everything and we sequence the heck out of it and we find out what's there, and then we try to predict from that information, what might emerge. This is where that is. There's a lot of interest in, using Twitter and news feeds and Facebook posts and media outlets. Those tend to happen once this has already happened and we're in an incipient outbreak. The idea is that what we hope to do is to sample things in this area. Okay, so how does it actually work? So this thing here is called an insectazooka, and this is me at our site trying to, and they were laughing at me the whole time because I have my nice, collar shirt on and I'm out here doing this. But there's mosquitoes that are sitting here that have fed on the humans and animals that are in this person's house. You collect them with this insectazooka, you bring them back to the lab, and this is what a mosquito with a blood meal looks like. You express the blood meal from this mosquito onto a preservative card filter paper. Then these can be stored either in -80 or at room temperature and then sent back to the lab. I have parentheses around that because what we don't really want to do is to have to bring it back to Colorado State to do the analysis. What we really want to be able to do is to do the analysis in the field, and that's a place that we still have to get to. Anyway, right now we're doing the stuff in the lab, so when it comes back to the lab, we punch these blood spots out and elute the RNA and whatever else is sitting on there, and then we extract RNA from this, and then we can either sequence it by next generation sequencing here, or if we know what we're looking for, we can use RT-PCR. This is a portable sequencer that we hope to use at some point, but we're not doing that yet. From PCR data, PCR information, we can get data on prevalence, and then we get more information from next generation sequencing. We can learn about what the viruses are, especially things that we don't know about. Okay, so when we were first talking about doing this, that was like kind of a neat-ish idea, but the question is, when you go through the process of taking a mosquito out of somebody's house and squashing the blood onto some filter paper and bringing it back to the lab and doing all this stuff, do you lose the ability to detect that? And so we've done a lot of work showing that after, if we take the blood and we put it on a filter paper card and then we store it, we lose a little bit of signal, but not really that much. This is data for West Nile virus and also for HIV-1. So the idea behind that is that we are interested in arthropod-borne viruses, but the idea is that mosquitoes are collecting whatever's in the blood, and that doesn't have to be an arthropod-borne disease. It can be HIV, and you'll see some other examples of the types of viruses that we can detect using this method in a second. So what we really want to do, though, is to be able to survey for things that we don't know about. And so these are just some really, really preliminary results from many years ago of our next generation sequencing studies, where we made libraries and we put them on the MySeq. And so what you can see here is that almost half of the data that we've got really is not very good. There's low quality, just a bunch of adapters, redundant sequences. So that's not very good. And then most of what else is left is mosquito sequences that we don't really care that much about because we're interested in pathogens. And then Anopheles gambiae mitochondrial RNA and ribosomal RNA. And so really, of all of this data that we get from next generation sequencing, about 1% of it is actually useful. So we were like very sad about that when we saw it, and we had to think of ways to make it be better. So there are two ways that you could think about kind of dealing with that problem. One is to either remove some of the junk that's in there that you don't want, and the other is to enrich for the things that you do want. And so over the years, we've worked on a couple of these things, and one of them is just depleting common contaminating sequences. This takes advantage that you can selectively digest RNA bound to DNA using RNase H. And so if we design DNA probes to common mosquito sequences, we can just remove them out from our pool of RNA that we're sequencing. Also, we've designed, in collaboration with several other groups, probes that we can use to pull out the sequences that we're interested in. And that can be for any of the known viruses that are out there or a selected set. So that's been a sort of major effort over the years, is trying to figure out the molecular biology of making this work better than it did when we first started out. So sort of with all of that in hand, one of the first things that we wanted to do was to see whether it would actually work in the field. So in the lab, it sort of seemed like we were able to detect things, and that we had some methods in place to make it work better. So the first study was done in Liberia, and this is in Lofa County. And these are the people who really did the work. This is Joseph and James and Nathan, and then local people there were also really important in us, in our being able to do this. But this was happening kind of during the big Ebola outbreak in Liberia. And Lofa County is where Ebola first emerged, and that's where we were working kind of as that was going on. So that was pretty interesting for those guys to be out there. But the idea behind this study was to go to a couple of villages in rural West Africa and do just what I said. So go in the morning and collect mosquitoes and squash the blood from those mosquitoes onto filter paper, and then in parallel collect blood from the individuals who are living in these dwellings and see whether there were matches. Okay, so this is some data from those studies. So from the mosquitoes that we collected, we detected GB virus C, which is a common virus. And if you look at this, this is the GB virus C genome. And if this thing is black, this is an area that's covered by sequences that we obtained from mosquitoes. And if it's blue, it's sequences that were recovered from samples that came directly from people. And so what you can see from this is that in this village, you get GB virus C in the same household from the people and from the mosquitoes that are feeding on those people. So that sort of makes you think that this is probably something that could work, at least for this virus, but it's not just for this virus. We saw the same thing for hepatitis B virus. This was present also in a village, and also in samples that derived from both mosquitoes and from people. We also detected Epstein-Barr virus. This is a common virus in African villages, and so these are the different villages and different sampling spots. Interestingly, we also found a mosquito that had pretty good evidence of canine distemper virus in it, and so that was kind of interesting to us because, canine distemper virus, you wouldn't really expect that to be in people. So the first question is, well, I wonder where the blood from that mosquito came from. So we were able to go back to the blood spot that gave us the canine distemper virus sequence and determine that that mosquito had happened to feed on a dog, which is also a kind of interesting One Health piece of this, is that we can use the mosquitoes because in many cases, and you'll see, you know, the households where the people live also have domestic animals, livestock, birds, and so forth. And so this is also kind of, you know, not as interesting as a person who's infected with canine distemper virus, but sort of proves again the larger principle that you can sample mosquitoes and thereby sample people and animals that are in the house. Okay, so now we're kind of moving into the current project. This is from a backyard in the region where we're working, and you can see the latrine there, and that there are pigs and chickens, and there were ducks here somewhere, and then back in this pile of stuff back here, it was really easy to collect a lot of Aedes aegypti mosquito larvae from back there. There were mosquitoes all over this place and all over in the house that was this backyard was from. So the idea is that humans and animals and arthropods intersect a lot in this area. So this is kind of what we're trying to do. We're trying to conduct vector surveillance and risk assessment. The first thing you got to know is what are the mosquitoes that are there? What are they capable of transmitting? How abundant are they? What are the things that are feeding on people and animals the most? This is an area where, excuse me, insecticide resistance is a real concern. In many cases, there are not great options for insecticides. And then this whole idea, and what the main bulk of the study is household xenosurveillance. And so the idea behind this is that we're enrolling 20 households. At enrollment, we're collecting capillary blood from everybody who lives in the household and then up to 5 of their animals. We also are aspirating mosquitoes from within the house, identifying them and collecting their blood onto these FTA cards. It's a preservative filter card. And then we're prospectively following the households, and then we're visiting each time that there are symptoms that develop. And then the idea is that using that approach, we can compare direct human surveillance to this idea of xenosurveillance. So this is just a little bit of data from enrollment, and we have a really great local team there, and maybe Valeria will tell you a little bit more about that. We got the 20 households that we were hoping to. They come from these 2 villages, and I guess I can just say that there's 46 kids and 52 adults that are the humans that are enrolled. There are a total of 89 animals that are also enrolled, and then we collected 667 mosquitoes at enrollment. 227 of them were blood fed, and those are samples that are waiting for us to work through currently. We've been following the households up since the end of August, and this is just a piece of the data to kind of give you a flavor for what this looks like. Some fever cases, rash, and kind of body aches. We have some instances of those in each of these weeks, and these are the household IDs here. And then we go back and we collect animal and human blood, and mosquitoes with blood and mosquitoes without blood, and those are all, again, kind of waiting for us to start working through them. So, you know, kind of where we're at with the project is that, you know, we have a lot of samples, like several, you know, times what I thought that we would have at this point, so we are trying to figure out how to prioritize which ones to go through first. Likely it's involve some sort of like a phased or tiered approach where we're working on the human samples first, and we'll be using next generation sequencing because we don't exactly know what to expect to find there, although we can make some guesses. I would hate to sort of miss something because we think we know too much. We're the 3 of us working on getting additional funding for the project. We've been really lucky to be supported by the CSU VPR's office and the One Health Institute for many years now. And we're just kind of trying to figure out how to keep doing this because we think that the site and the people and the methods are all really promising. Ultimately, as I alluded to earlier, what we want to be doing is doing this stuff in the field. It's not especially useful to the people there if we have to kind of save things and put them in the freezer and then bring them back. And so in the next year or so, hopefully we'll kind of be getting towards that. That's kind of the next technical hurdle that we have to go over. So I'm going to stop talking here. I've mentioned thanks to the One Health Institute in the VPR's office, Center for Global Health and the Centers for Human Development at CU, and then the university that we collaborate with in Guatemala, and these are many of the people who we work with. Yeah, you're welcome.
Well, here you have the same picture that Greg showed you before, and the idea is that basically it's the same thing that we want to reinforce. It's like how people live in very close contact with animals and with mosquitoes. You can also see from the pictures that nobody is wearing shoes. Then there's the latrine in the back, the tire collecting mosquitoes. So you can see that there is this interconnection between humans, animals, and the environment. That's why we think that that's the best approach to address health issues in the region. Okay, this one? Isn't this one?
The yellow one's the laser.
No, it's this one, right? That's right.
Maybe Greg broke it.
Well, I can keep going on. Oh, I can, yeah.
All right.
Well, oh, I can do it.
We're way
No,
ahead of
that's fine.
ourselves.
That's fine. I can do that.
Sure. All right, let me
Yeah,
get- you keep talking. I'll find your
Oh,
spot.
okay. Thank you. Well, and the next slide, what it's going to show you is research from Dan's group from the Center for Global Health, where they, oh, it's before that. Yeah, it's this one. The next one. No. Well, their research showed that there are a lot of animals in the region. There's a high prevalence of domestic animals in the households. And in that table that I'll show you later, you can see that how many animals are in each household, inside the house, in the yard, and in both. And in most of the houses, this one, yeah, thank you, Dan. And so you can see that in most of the houses, the animals circulated in between, in the house, outside. Every like the animals are inside, outside. They are very, very connected. So that's why we think, well, they started working with humans first and the mosquitoes, and then it was appropriate to start testing animals and start evaluating the pathogen burden, or which type of pathogens these animals are harboring, because they're in very close contact with humans. And so last year, we've been writing on several grants, and at the end of the year, we were awarded one health grant from CSU or VPR. That's the one that Greg was talking before, just to measure transmission dynamics of mosquito-borne diseases. And then at the beginning of the year, we saw another call for a PRECIP, so we decided to apply for that one too, and we obtained that one. And the idea was that since we were going to be in that region, and since we were going to collect blood from animals and from people, why not to use that blood to test for other pathogens? So that's another tropical, that's a tropical area, as you can see. So ticks are prevalent, fleas are prevalent too. So we decided to just collect some ectoparasites and test for selected ectoparasite-transmitted diseases. So this summer, in August, we went there, enrolled 20 houses, and those samples were used for both studies. First, for the PRECIP, that's the ectoparasitic transmitted study, and then the One Health, and then for the One Health being like baseline data. And we collected animal samples and human samples, so we have 98 samples from humans and 89 from animals, and then we collected ectoparasites, fleas and ticks, from 18 dogs and cats. The chickens and the pigs didn't have anything. It was mostly dogs and cats. And this is, again, how the One Health study is going on, and Greg shows something similar. And this is how we are reporting results every week. This is only for 4 weeks, but that's what happened. Every time somebody in a household had fever or rash, a nurse contact them, goes there, collect samples from the humans in the house. The veterinarian goes there. We have a local veterinarian working there and collecting samples from a selected group of animals. And they collected mosquitoes, and everything is recorded. So up to now, we have finished the collection for PRECIP. We are still collecting samples from the One Health study. To bring the samples to the US, we require permits, and we require CDC permits for dogs, cats, and ectoparasites. And then for poultry and swine samples, we require USDA permits. We have some of them approved, the USDA permits, and then the CDC permits are approved, and the USDA permit, we are still waiting. It just takes longer. And we were expecting to receive a shipment in this week on the next one with a selected group of samples. And then at the end of the study, when we'll receive all the samples, the idea is to process all together just to make it more efficient. And then, well, this was the first veterinary study done on the site, so we have some challenges. But we contacted the veterinarians from the UVG, the Universidad del Valle de Guatemala, and they have tons of field experience because those are the type of studies that they do. So through them, we hired a technician who was very helpful in collecting samples, knowing how to address people and how to engage them. Then another problem that we had is that then we were going to come back to Colorado, and we need to keep collecting samples for the One Health study, and there's no veterinary, any clinic in the region. So there are only 2 veterinarians in the closest town that is between 40 and 50 miles away. But who are the ones that are collecting the samples and collaborating with us right now. If not, it would have been very difficult to do this. And then we are, as you can see, too many people, different countries. But the group has been working, as Dan said before, since 2011. So they have been, the team is already established. And the communication is actually very good. We have weekly phone calls. We just have WhatsApp messages all the time for daily questions or things. And so we're very happy with that. They are wonderful to work with. And then our major challenge is like, we can see that there are many needs in the community, and we really like working there. So we hope to be able to continue working there. And then, our goal is not just to do a few studies, it's more like to continue in the way that Dan has been working, and Greg too, and then just see if we can include some veterinary surveys or more animal care studies. And this has been done by these 2 grants that we obtained. Then Greg submitted an R21 last month. And then we hope to keep, we're still working on submitting more grants and keep working there. And then, again, all the people who work from Colorado, from the CSU, from CU, from the Universidad del Valle in Guatemala, from people in the site who are collecting the samples and doing all the following of the One Health study right now. So they are all this group of people that are wonderful to work with. And then I just want to acknowledge for the animal part this group, because they have been wonderful in not only collecting the samples but coordinating everything, because sometimes you go to the houses and they said, oh, a chicken was eaten, so it's not in the study anymore, so you have to find something else, or the dog The dogs are stray, so they are in the house whenever they want. So you go there and there's no dog, but maybe tomorrow morning when he comes for breakfast, he's there. So they're wonderful in coordinating everything, and nobody gets upset because the dog is not there. So I am very thankful to all of them, but especially to these 4.
Thank you. So I had prepared a question for each one of you, but it's open for questions and answers, and
Do you want us to sit up here?
maybe easier. Hey, and good handwriting.
Yeah.
My handwriting was perfect, wasn't it? Okay, and I mean, you can choose who answers this. So, I mean, it isn't easy to put together teams to work in One Health. So this one, you can decide who answers. What would you say are the advantages of working in a team that combines human health, animal health, and xenosurveillance?
I guess I'm happy to answer. So I think we collectively, as a scientific community, recognize that you really can't study human health in a silo. I mean, you saw the pictures. These people are in constant contact with their environment, whether it's their affected latrines, whether it's the animals, whether it's the mosquitoes. And the factors that play into human health, you're not really understanding the full picture if you're not also looking at the other aspects of their exposures and their risks. And so I think it was clear to us for many years at the site that we needed to try to involve these other aspects of health. And it wasn't really until we just connected with Greg and Valeria to kind of start thinking ideas through. And I guess this is kind of getting a little tangential, but in this example, we had all these ideas, and sometimes you just need to take the first step. And this grant was a great, easy pilot first step that's now leading to other things. And so I think when you have people in different fields, it can seem vague in what you really want to do, but if you just kind of go out there and take your first step, that can help get the project going. And now we have a direction that we're taking things.
Thank you, Dr. Olson. Now for Dr. Greg, because I never know how to pronounce your last name.
It's like evil with a B.
I'm always worried to pronounce it in the wrong way, evil, yeah, it's in Spanish. So I've seen that, and I've seen it from the 3 of you already. They are very interested in local capacity building. You all brought up that in your presentations. Could you please speak up a little bit to the entomology laboratory that you're building there and how you foresee building that local capacity?
Okay, so I can tell you what's happened. I'm trying. Okay, so how do I start answering this question? Ultimately what we want to be able to do is to do the relevant experiments in the place where the work is happening, and to train people there. We bring into the lab as part of this project samples that you maybe don't want to have in your diagnostic lab because they're dirty samples, and the type of equipment and the type of stuff that you need to do to start your studies of mosquitoes is different from what you need to do PCR for a diarrheal pathogen panel, for example. And so, so to develop the entomological capacity in the site, a trailer, a shipping container was donated by Banasa.
Yeah, paid for by Banasa and delivered by Project Cure, which is in Denver.
Okay, so we were really lucky that we got this facility, sort of the structure of it there, and then that's been converted into basically what's a field insectary. And so there's 2 parts of the container. The back part is not climate controlled, and the idea is that if you wanted to do studies with Aedes aegypti mosquitoes, you could bring larvae, you could bring eggs in, and you could rear mosquitoes in that part of the insectary. The other part of the insectary is climate controlled, and that has a desk and some dissecting scopes and a refrigerator and so forth. It's like a little lab, and so you can use that part of the lab to identify your mosquitoes and do the parts of the experiment that you prefer to be in a temperature-controlled environment for. We have developed mosquito identification keys for the local workers who are there, who are, brilliant but not entomologists. So we've tried to give them some sort of basic training in how to identify mosquitoes. We haven't really gone there with rearing mosquitoes yet because we just haven't had the funds or the reason to do so. But I mean, we're really excited because that's a really nice field insectary, and we just sort of need a project that would give us the reason to do that, and then we'll do it.
Thank you, Greg. Excellent. And Vale, would you like to, I mean, and the three of you also recognize there's been a lot of support from CSU and CU. But in the ideal world, if you could ask for things, what would you need from both universities? And we are not going to abuse Dr. Dr. Rudolph Allen here, but Alan Rudolph here, but and you can talk about CU probably.
Well, from our veterinary point of view, I think that we can do a lot, and I think it was a good start. These studies are a good starting point, because when we went to the houses, I asked people, "Have you seen a veterinarian? Have you ever go to to a local place even to buy, dewormers or vaccines or something?" And the only thing that everybody told me was that they only have the rabies vaccine given by the Ministry of Health, that when they vaccinated everybody, but nobody has seen a veterinarian. And they said the question was like they were far away and everything was expensive. So, but not because they didn't want to. So I think there is a lot of potential of things that we can do in order to contribute to the health in general. So the way I see it is like it would be nice first to start maybe talking to the community and telling the things that you can do in terms of improving animal health, deworming. Also, the other question I was asking them was if they would be willing to do spay or neuters in their dogs and cats. And they all agree. They said the only reason is just an economical reason, not because we have anything against that. So one part will be just establishing kind of like a preventive medicine program in terms of health. Then they have a lot of chickens that it's like a backyard poultry that we can also kind of, I think we can just establish some sort of basic preventive medicine in terms of animals. And then the other part will be to keep doing our One Health research projects to see. I'm very interested. We were talking to Dan a while ago about the diarrhea. Just to do to include animals in the testing for diarrhea. Also maybe also the soil, the water. Something has been done with the water, right? So.
And would you like to add something?
Yeah, and I think one of the difficulties with doing research in a resource-limited setting is that grants are happy to pay for materials to do your testing, for salaries and things like that. One thing they don't pay for is infrastructure. And so that includes like big diagnostic assays, like these mini-ion next-gen sequencing assays you can put on in the lab down there. And so we were fortunate to get, it's called the Strategic Infrastructure Research Council or something, CIRC, it's a grant you can receive from the University of Colorado that provides money for infrastructure at a promising research site. And so they gave us funding to further develop our site. And so that included building some housing. It included money for solar, actually, because the power supply there is very inconsistent, and that will fry your equipment if you don't have a steady power supply. So with these -80 freezers, we had to install solar. And then it also provided some funding for this entomology lab. And so I think one of the things we always struggle with down there is you need to have funding in order to get infrastructure, but you also need to have infrastructure in order to get funding. So it's kind of like this never-ending cycle. And so for us, one thing we're trying to think a little bit more about is what are some of these areas that we really need to beef up a little bit in order to get bigger grants. And so one of those would be, we have a couple of changes we can make on our the entomology lab to kind of make it kind of ready for prime time. but also things like the lab, like Greg mentioned, having our next-gen sequencing assay on site. Each time we send samples back to the U.S. on dry ice, it costs $2,500, and a grant will pay for that. But if you just send 10 samples back, you're already kind of getting high enough costs that it's almost worth it just to get the assay down there. And obviously your results come back faster, and capacity building and all that. So I think for me, the infrastructure thing is a big one. The personnel, these people down there are hungry for opportunities. Like our nurses and our team are all really great, and they're taking master's classes at the capital city, which is 5 hours away on the weekend. I mean, they're really want to advance themselves. So that's not a problem. It's more about these kind of infrastructure things that grants don't typically pay for. So we're trying to get CU to hook us up with some more money.
Can I just say one quick thing? So I think we've been really lucky to have been supported this far to get as far as we got, and that includes support from DoD to get kind of the molecular biology pieces of it together, and this is kind of going into the field, and I think kind of the next step for us, which would be in the next 2 to some number of years, is I think we really need to be able to show that what we're doing there is transformative from a health perspective, and we're not really there yet. But I think that we're kind of poised to be in that place. But I think that in order to get there, we just need to have the resources to do the kind of study that we're doing right now, and I think that's kind of what we're all kind of working towards in different ways.
I'll just ask one more question, and then it will be Dr. Alan Rizzo's turn. Doctor, we are very briefly, I know you've been very interested in looking at how to search for probably Department of Defense monies, since this is an area where the migrant population is coming from. You had some ideas on a type of study that you were suggesting to do in that area, and then we'll pass it to Dr.
Well, I definitely don't want to talk about defense money. That makes me very nervous, especially in situations where you have a lot of migration happening. But this is an area where a lot of migrants cross the border into Mexico. It's a known corridor, and I've in my past done a lot of research on migration patterns and unaccompanied children crossing the border. A lot of the workers that work in sugarcane and other industries are internal migrants too. Within the country. And so they're coming from other regions in Guatemala where they might have their own home and farm, and maybe due to drought and other conditions, sometimes climate-driven, are becoming workers in these locations. And then also you have people leaving out migration up through Mexico towards the United States. So I don't know what all you wanted me to comment on, but it's just an underlying background issue to this location and some of the issues that are happening. So it's something that I was thinking a lot about as not related to this study specifically, but as maybe something else to build out as an additional layer to look at climate and migration and how those issues affect not only this population of workers and their families, but also their health and other issues. Yeah. Oh, sorry. Yeah, my name's Betsy Weir. I work at the One Health Institute with Magdalena. Yeah. I have my background's in sustainability studies, and also I worked in international development, primarily in Latin America for many years. And I lived in Guatemala twice. Not once, but twice. So love it, and hope to continue working with this team in some different ways, and maybe just brainstorming ways. Yeah, thanks. Alan, go ahead. And then we can pass that around for questions.
Yeah, sure. Thanks. First, I was just saw Sue Vanderwood, so she wanted to extend her apology. She wasn't able to come, but I know she's keenly interested in this project as sort of the new One Health director. I want to come back to your comment, Greg, because, obviously, as you know, our office has also agreed that this approach could be transformative. But now applied in this context, I'd love to hear the teams kind of thinking about how that transformation takes place. So it seems to me what you've done very effectively over the last few years is show proof of concept that we can go in, use the mosquito as a sentinel for an area. And now I think appropriately you're showing how that crosses health considerations in the different channels of humans, animals, and the environment. So clearly, I think further the next few years, as you suggest, I think will plant that flag in a community where you will do a more complete assessment, and I sense that that's kind of what you're about. What I don't know happens is once the assessment is done in a region like this, what happens then? Who are the officials that take the information and do what you're suggesting, change the health of the community? Because as Dan showed us, the burden is already so high. The influences on that burden are known: water, the floods, cisterns, many things you pointed out. So I'm wondering what's in the team's mind about how to take the assessments that you're doing and will do. I think we voted with our feet and believe that you'll eventually get extramural support, and this will continue to be an area that we learn even more about in terms of sampling. So that's one question. The transformation and assessment that you're doing crosses boundaries in which the actual application of public health in that region is not clear to me. And then the second question, because you started out with the left of boom discussion, and what I think also is enabling about what you're doing is not necessarily the assessment, but the dynamics or the plasticity of the equilibrium in the region, right? Because the boom is really all about how does this thing go out of equilibrium? What triggers the spillover? You mentioned, first it goes into a few humans, the R naughts, and they're the viral shedders, and maybe that's the flashpoint. So I guess the second question I have is that, are we anywhere near in the development of the tools, surveillance, to actually look at the evolution of any one pathogen? Are you able to yet sequence enough out of any one pathogen to see its change over time? Because it seems to me once you have the assessment tool well in hand, what you then have the ability to do is look at the dynamics, right? What is the evolution and the plasticity of the burden that's going on? And can you see something that tells us about when the tipping point, or what about that disequilibrium can we be predictive about? So I'd love the team to talk more about what Greg said. How does it view the transformation playing out, assuming the success of assessment, and then can the assessment lead you to some plasticity, which is kind of the holy grail of viral evolution? Can we predict when this equilibrium of many things in a high burden area somehow goes out of disequilibrium?
Is this working? Yes. Okay, thank you. I will tackle the first question as a global health person and part of this team. The way that you'll focus with this type of research is wonderful to have first a group of selected researchers that are looking at the problem from all the angles. The way to tackle this in global health is that you work with the government. So we'll have to be working with the government, the Ministry of Health. We have to work with the water people. We have to empower the local authorities, being very difficult in Guatemala because of the political changes that change very often, and they change the cadres in Ministry of Health. They don't keep constant technical cadres. That's a problem. I've worked in Guatemala as vets in the past. Do you have several potential partners? The other thing is good with the CU is that they are already a center for WHO. So they are recognized, and we work with Pan American Health Organization, WHO, to bring in other partners. The other angle that they have, which is very interesting, is that they are linked to the private sector. In Latin America, to make change sustainable, you need to engage with the private sector due to the instability of the governments. So I think there's a very good possibility, but again, you have to tackle, as you were mentioning, several problems at the same time. Once you identify the causes of the pathologies, and you know that they're there, you have to do through public health changes in water systems. You have to work in development for the children. I mean, it's going to require action that it can be done, but it's bringing together many, many partners. And it's not the government alone it's hard for them.
Yeah, why don't?
So yeah, I totally agree. I think as researchers, sometimes you can get stuck in this problem of describing a problem or a health problem to death without actually doing anything about it. That's it's the whole basis of the implementation science. And I think our site, we've been running really for about 5 years, and I think we're finally starting to really have a good sense of the different disease burdens in the community and the risk factors and everything. But I think you're right. I think now we're at a point where we need to start focusing a little bit more on interventions. And just kind of backing up a little bit, I think, to some extent, actually just getting research grants and funding down there actually improves health in this community. For this Zika study, they conducted over 2,000 developmental assessments on these kids and referred them as appropriate. But it also kind of opened up this box, the lid on the box, and shows that there's huge problems in this community, and we're starting to kind of have a better sense now of what are the priorities. So diarrheal disease is huge, but also dengue, Zika, arboviruses are another kind of big problem, and there's many other problems too, worker health, those types of issues as well. And so I agree, now it's time to really, as a group, for us to start thinking about where to go from here. And I think to some extent we can use the data from our research studies to drive what interventions we want to do. Valeria mentioned the study about enteric diseases, and there's been some very disappointing studies, WASH studies, so water, sanitation, hygiene studies they had these huge interventions, thousands and thousands of children, and in the end, there was no significant benefit to these multi-million-dollar studies. And I think that makes us really kind of understand that we don't fully understand how pathogens are transmitted, how they impact health, what are the long-term outcomes. And so not on a general scale, I think some of our research can still address what are the real causes of this. But even on a local level, how can we look specifically in this community? Maybe Campylobacter is being transmitted by dogs in the household, and One Health study is a great way to understand that. We can then use these community health groups like Creciendo Sanos, for example, that first slide I mentioned, to try to deliver that message. I think one advantage that we have in this community is that this is a rural area, and our footprint has always been in the community. We're not just some centralized hospital waiting for sick patients to come to us. All our programs are out there in the field, and over the last 5 years, we've really got buy-in from this community, and they respect what we do. Initially, they were suspicious. Anytime you have an outsider coming into Guatemala, there's reason for them to be suspicious, but we've established this rapport with the community, and I think now we can use that rapport to kind of deliver the highest yield healthcare messaging we can to that community, whether it's enteric diseases or arboviruses, and what are we picking up in the community at any given time? How do you prevent mosquitoes? And we've worked with the Ministry of Health. They have little health outposts throughout this area to help reinforce that message. And so it's certainly not easy. The real solutions are eliminating poverty, getting good water. But as taking kind of baby steps towards that, the first part is using the data we're collecting to really drive the messaging and hopefully taking it further, the further out we can get. So I think it's an important question, though. I think as researchers, we often get caught up in getting papers published and not thinking of the next steps and how does the local community benefit.
So I'll maybe try to address the second part of Alan's question about, where do you, just let me rephrase it to make sure that I understand it. So, can you use what our overall kind of One Health plus whatever else we're doing approach to kind of understand the tipping point between kind of this ecological, for lack of a better word, chatter, and then a big problem, and by the time you're at that stage, it's over. You missed the chance. So I think that, part of the reason that I mentioned, I think twice in the 15 minutes, that we need to get diagnostic testing into the field and away from here. That's like step number one. Until we can do that, there's no possibility to do that. And so that's part of what I'm referring to as this idea of being transformative. In order to do that, you need to have that piece of it in the community too. And I think that we have now moved beyond proof of principle, but to get to that next stage, it's another investment, and that's hard to do. So that's what we're trying to figure out right now.
Good morning. Thank you all for being here. So my first question is for Dr. Olson. You were just speaking about suspiciousness in the community. So in regards to I believe it was the pediatric Zika study, I'm curious about the participant receptiveness to the psychologists, opening up about what might be considered intimate information.
Yeah, that's a really good question, and it's something we thought a lot about ahead of time. How do you approach a community? And sometimes, I think maybe a good way to look at it is they're in this box, they don't have much exposure to other, higher income settings, and they don't necessarily appreciate that a child that can't talk by the time they're 3 is abnormal. And so they're like, all our friends are like this. So how do you come in as an outsider and tell these people that, collectively or individually, that these children have major developmental problems, and we don't really have the resources to help prevent malnutrition in the community, but here are some ways in general, to kind of improve your health. So we were very conscientious of that, and that's an approach we take with all our studies. We do a couple things, first off, for any study to kind of make sure we're approaching things culturally appropriately. One is we have a community advisory board, so for any study, we have our Colorado IRB, we have our local Guatemala IRB, which could be a local university or the Ministry of Health. Then we have this very local group that we basically run every study we do by them and get a very local perspective on how people will receive this idea, what are some ideas to get engagement, and how can we share the results with the community. We also recently started a program where I'm the research director for the site, and any study now has to come through this system where the people doing the study have to tell us how it's going to benefit the community and how are you going to share your results with the community. And so the goal is just to maintain the utmost transparency in everything we do. And it's taken years to kind of really build that trust, but having that transparency is a really important way to do that. And so with this developmental testing results, one thing we've done is we kind of did all those steps, but we also are working with local Guatemalans who are actually doing the testing. Our neuropsychologists from CU, actually 2 of them, Amy Connery and Allison Colbert, are completely fluent Spanish. Amy spent 2 years living in Honduras, so they have a general sense, but really having the local psychologists actually carry out the testing and then have auditing and feedback from the kind of more experts has been a nice way to really get that engagement. And the more, after doing a couple hundred of these, they really developed a good flow of how to interact with these patients and kind of have some suggestions for things that they could do at home without, making them feel bad or, going too far, basically.
Definitely.
One of the psychologists from Guatemala is doing an MPH now, if I'm
Yeah. So one of our psychologists is at our center now. She's doing a global health fellowship, which we offer, and she'll be here for 2 years. She's doing an MPH study. Our local study coordinator for the Zika study is actually doing her PhD at Columbia on implementation science. And so we are really trying to kind of build the capacity through training locally. And unfortunately, some of that means is that they have to come to the U.S., but ideally, eventually return to the site or stay involved and continue their work there.
Cool. Thank you. I have more questions. If no one else has questions.
Does someone else have a question or? Okay, I'll come back to you.
Good morning. Thank you guys all for being here. My question is about the involvement with the university. How far away is it from El Trifinio?
Yeah.
And, and you mentioned it and you were very forward in your thanks for the university, but I was wondering what the details of that relationship are like and how you guys are utilizing the university there.
Yeah, comment. I think anytime you do research in another, especially a low-income country, it's really critically important to involve local researchers. And Universidad del Valle de Guatemala, that's UVG, they've always been the kind of main public health, epidemiology, veterinary, entomology university in the country. They're based in the capital, which is about 6 hours away. And we've always had this connection. We have local kind of MD investigators down there that are affiliated with the university. And so we've always kind of interacted with them on various levels. And so they've just had a strong interest in collaborating with us for for a number of years now. And it depends on the study how much they're involved. You know, not every study, you know, the Zika study, for example, they weren't as involved because it came, you know, through the university and was local, and they didn't have a lot of expertise to bring to the table. But for the One Health studies we're doing, they're actually a great partner. They have veterinarians that are already doing field work in animal health. They have entomologists who are doing, you know, malaria work and dengue work, and so, and, you know, virologists, and they have a good diagnostics lab too. So it's great to have them because we're kind of a small mom and pop shop out in the boonies, and they're this big university in the capital city, and they have the expertise to help us on, you know, still have kind of like a local perspective. And so when we do when we're trying to figure out a diagnostic platform for our site, we work with them to help do the training and teach us. There's a red cap person who knows a lot about red cap there, who does, like, he comes down and visits and does trainings every month with our data managers at the site. And same with epidemiology in a lot of these areas. So it's nice having this like local meeting, like still 5 or 6 hours away, but within Guatemala to kind of help maintain that capacity.
As a follow-up to that, did your relationship with the university happen at the same time as you're developing the site in El Trifinio, or was it before or after, or what was the timeline for that?
Yeah, one of the things I'd say is I should have given a big thanks to is our site director is Edwin Asturias.
Mm-hmm.
He's pediatric ID, infectious disease also, but he's from Guatemala and he's from the region, and he played an integral part in setting up the site. He has had a faculty appointment with UVG for years, and so he was our kind of introduction with them, and ever since we've started, they've been involved in the site. So it's good to kind of bring your local partners in really early, and then not only do they have a good perspective, but it also gives them buy-in to the site itself and an interest in maintaining and improving the site.
Maybe Dr. Scorza wants to add a little bit about the veterinary, because they've been also very helpful for the veterinary component.
Yes. Yes, I think you worked with some of them before, and I met them through the CDC. We wrote other proposals in the past about enteric diseases, but we never got them. And then we've been in touch, and they are the ones who, you know, they do this type of studies, like the veterinarians do this type of surveillance for different diseases, from rabies, for pretty much everything that is zoonotic. So they have the expertise, and the technicians are wonderful. And the other thing they have is like they know how to address people. Like I speak, you know, Spanish, and I'm just fine. But I remember being with Ramon, the technician. He does that as a living. This is his profession. So the way he addressed, you know, the community, like, hey, what about, you know, let me talk to your wife. Maybe she's interested. We sample the chickens or this type of things. And they were wonderful in providing, you know, all the logistics of how to do it. They even, you know, we talk when we came back of how we did our sampling and everything, and the things that we can improve. Like some things they said, oh, I can buy some of the supplies here, so you don't have to bring them for the future, or and they are wonderful in collaborating, the veterinarian and the technicians I work with. So.
Hi. So my question is, how do you address the environmental sector of One Health? Because it's always a focus. We always focus on animals and humans first, and it's obvious you would also take into the importance of how the environment that they interact in. But what do you do to specifically address the environmental aspect?
I will suggest that you give the microphone to our colleague from environment.
Oh, actually, I was going to say maybe you guys should comment on the water and soil. I don't know if you have enough
it's going to be researched too.
Yeah.
We don't have the funding, though, to do it yet.
Well, I can give a couple of examples, I guess.
Yeah.
So, so if we get funding, that the study that I've mentioned a couple times is, you know, focusing on animals and humans, but with enteric diseases, you really do have to study the environment. So, looking at you know, the sink in the house or the different environmental sampling points, and find out where the pathogens are actually being transmitted from the animal to the humans, because there might be a link there, but you also want to understand how that's happening. And so that's an example of where an environmental sample would be really helpful. Another example is this idea of this Mesoamerican nephropathy, and understanding how kind of the environment as a whole affects health. Liam Newman is a faculty member on our campus, and he's been leading this foray they study this. They work at a separate plantation for sugarcane, and they've done the studies that show that people's creatinines, their kidney function, gets worse just over a single workday. And when you do these interventions to provide hydration and electrolytes, their kidney function improves. And they actually have also brought in the whole productivity thing to show the company that these workers' productivity improves when they're well hydrated. So now they've made an economic argument to show that these workers should be giving hydration and electrolytes and breaks during their day, and that's improving the economic output of the workers. And so taking that environmental context, and how this might even change over time with climate change, for example. So taking that environmental context actually has led to a specific health intervention that is improving the health and the workplace environment of these workers. So there's a couple of examples, but I agree, you can't really ignore the environment. One of the problems with One Health is sometimes you get so vague that you're just like, well, everything is part of One Health. But I think it's important to kind of bring in these different factors and where they're relevant and understand how they're playing into everything. Absolutely.
Okay, Dr. Ebel, I have a question for you. So you quickly mentioned insecticide resistance in the community you were working in, and I just kind of wanted to get a feel about what the community felt about insecticide application, if you have any of that information, and then alongside that, whether there's any education about removal of breeding sites.
No. So the answer to all of those questions is basically we don't really know very much about it. Most interventions directed against mosquitoes involve some sort of insecticides. Environmental modification is something that you could think about. That's quite a bit more complicated because of the sort of dispersed and small nature of the vessels that are kind of giving you the mosquitoes.
Yeah, of course.
So we haven't done anything about that yet, which is why I didn't say anything about it. The approach to assessing the extent to which insecticide resistance is a problem there, given the samples that we have currently, is to look for molecular markers of insecticide resistance. Susceptibility and resistance to pyrethroids is sort of conferred by a few fairly well-known mutations, although I would argue that we probably don't know all of them. We can get a pretty good handle on it through some molecular assays. Part of the reason for having the insectary down there, however, is to measure that directly using things like bottle assays that I'm sure that you're quite familiar with. But so that's kind of thing number one, is to assess the extent of the problem. It's in the middle of an agricultural plantation, so I'm just going to guess that I'm pretty sure there's an extensive insecticide resistance in the mosquitoes in this area, and managing that and figuring out how you can use insecticides, if at all, in this environment, I think is pretty interesting, but is going to end up sort of falling outside of what we can do in kind of the current thing. But.
Great, thank you.
CDC has some funding for us to work on that.
I know, that's what I'm thinking over here. I'm like, so let's talk.
Yeah.
Any other questions?
Thank you very much to all the panelists and to the audience.